of 62 plants obtained from the various repository to identify putative start domains across the plant kingdom. The significant hits are processed again to remove false positive and redundancy. Domain structure analysis was carried out to explore additional functional domain associated with the start domains and their spatial arrangement in proteins, which further help to classify start proteins into different structure classes based on their domain arrangement. These different class of start proteins were then functionally annotated based on their gene ontology, thus providing an overview of functional divergence for different structure classes. Further, gene structure analysis was carried out to understand exon-intron patterns for different classes of start domains. So, genome-wide identification revealed that gene family size of start domains vary enormously across the plant kingdom, ranging from 1 to 5 in lower plants like chlorophyte to amplification of up to 60 in some of the genomes like soybean, triticum aestivum, and Helianthus anus. Further, in general, the gene family size of start domains gradually increases from lower to higher plants as the taxonomical hierarchy increases. So, domain structure analysis was performed on non-redundant start proteins to identify the domain structure patterns. Based on domain structure analysis, start domains of plants were classified into 6 major structure classes, just like Mecla-associated start HZSM, HDB start HZS, HS, PSD, SD, and minimal start. In addition, few peculiar or minor structure classes were also found in some of the plant start species. Among the minimal start proteins, start domains are not associated with any additional functional domain. So, the domain structure analysis revealed that start domains were mostly found to occur with the homeodomain transcription factor and comprise almost half of the total start proteins. The distribution of these 62 among these 62 plants species shows that in lower plants like green algae, mostly the minimal start proteins are found. In contrast, in higher plants, start domains are primarily found in multi-domain proteins and mainly associated with the homeodomain transcription factor. Further, the gene structure analysis of different structure classes of start coding genes shows that the size of start genes varied from 4 to 10 kV. The homeodomain associated start genes showed moderate uniformity, in which size of start genes varied from 5 to 6 kV, while the PSD and SD class showed slightly longer in size and it's varied from 9 to 10 kV. Most of the minimal start genes was found to be in range of 3 kV in most of the cases. Further, the gene structure patterns of these 6 structure classes showed variation in terms of number of exons encoding full-length start proteins and start domain regions. The representative image for gene structure analysis and exon representing different domains of start protein is illustrated here. Finally, the gene functional annotation of start protein shows the involvement of start in a wide range of biological process and molecular function, but are mostly associated with essential processes like cell differentiation, development, lipid and DNA binding, transcriptional regulation, and stress response. Finally, all data sets were compiled to construct a wave resource prediction tool named as a start finder tool or a start fit. A start fit is a platform for identification of start domains in new protein sequence as well as comprehensive online repository of start domains for 62 plant species. It is a multifaceted web resource for plant origin start domains and provide dynamic search options and visual inspection. The interface of a start fit is divided into 3 parts, mainly the browse menu, search option, and a start prediction server. The browse menu allows user to navigate a start domain containing proteins among 62 plant species. There are 2 options provided to browse the species, namely the taxonomic view, in which species are arranged in hierarchical order, and then the species table view in which plants are arranged in alphabetical order. On clicking a specific species name, a tabular view is open in which the accession ID of identified start proteins from a specified species are listed along with their sequence feature description and gene ontology terms. Here, options are provided to download the tabular data along with protein, domain, CBS, and gene structure. On clicking, accession ID of on a sequence page opens, which allows the user to see the ID-specific sequence feature and their protein, domain, CBS, and gene sequence along with their domain organization. Further, domain pages are linked to other widely accessible platforms such as IntelliPro, NCBI, UniProt, etc. Further, the search option enables custom-based search based on keyword and sequence. The keyword-based search option can accept domain's name, geoterms, and geo-ID in which user can select one or more species. The sequence-based search option further enables the user to enter or upload protein or CBS sequence in FASTA format, in which user need to select desired species. The search result is displayed in tabular format and made available to download. Further, the start prediction server enables user to predict start domains among their sequence of interest. The same HMM profile which was used to construct a pipeline for identification of start domains across 62 plants has been embedded into prediction server to predict the start domain for user-specified sequence. The outcome of a start prediction are in the form of list of accession ID, protein sequence, domain sequence, alignment. The user can view or download all the data and results. The predicted protein and domain sequence can be further analyzed at various universal accessible platforms such as IntelliPro, UniProt, Conjure Domain Database, NCBI, and this option will further help user to investigate the sequence feature. So, to investigate the sequence feature, family, superfamily, and co-occurring domains. Overall,
Nihan, we couldn't hear the last part.
I think this was the last part. It's ended actually.
Okay then. So maybe there are small ending part that we missed.
Yeah, probably.
Is there any question? Or our presenter is here or not?
Yes, I'm here, ma'am.
Yeah,
I have a question.
Yes, sir.
Beste, could I ask the question?
Yeah, sure, please.
Okay, thank you. So Sanjeet, right?
Yes,
thank
sir.
you for the presentation. Quite comprehensive. I'm curious, like, how does this really compare with PFAM, ProSite, InterPro? You mentioned you use them too, and CADD, for example. A lot of this is already available. I think what you've done is you pull it together and you have kind of nicely organized the information.
Yes, sir. Basically, this is the whole comprehensive within a genome. So we can say that Arabidopsis has 35 start domains. Like I have taken the full genome.
Okay. So in terms of novel, go ahead.
Yes, sir. So this is this is redundant sequence. Like PFAM having within the species, PFAM have many entry from various database. So I have I have taken from the whole genome and then I have run my own profile.
I see. So do you plan to do this for all the other domains? You can just turn the crank and repeat this for all the other domains.
No, I am basically focusing on a start. Otherwise, I will HD is already known. There are so many papers. I am also focusing on pH start domain, pH domain basically. In plants, pH domain is not explored so much.
I see. All right. Thank you.
Thank you, sir.
Thank you for the presentation and answers again. So I think we can continue with the next presentation.
Okay, the next one is GNet-CAC, gene expression-based CAC pathway grouping and scoring.
Hi, I am Fatma Özdemir. Our study is GNet-CAC, gene expression-based CAC pathway grouping and scoring. In this study, we introduced GNet-CAC. It is a new method that scores and ranks CAC pathway based on gene expression data. Scientists and doctors use gene expression data for several purposes. For example, to understand disease mechanism and decide on patient treatment plans. Large gene expression data sets on diverse diseases are produced. The high dimensionality of data and the small size of sample make it difficult to interpret the data. Most of traditional gene selection methods are not adequate. Recently, new computational methods became popular. In this study, we use 13 human gene expression datasets and CAC data. CAC data contains 331 CAC pathways. We utilize NIME to test our approach. GNet-CAC uses a set of CAC pathways as a feature to perform classification. GNet-CAC produces performance metrics and a list of important CAC pathways for disease under study. We tested CogNet, MATE, and SVM-RCE. GNet-CAC follows a similar approach with a MATE and CogNet in terms of ranking and grouping of genes. CogNet ranks each CAC pathways by making cross-validation. Here, we integrated CogNet and MATE, where the process of the ranking is implemented on the groups generated by utilizing the biological knowledge in each tool. Furthermore, SVM-RCE utilizes K-means clustering algorithm in order to group the gene into clusters. Therefore, groups are linked with the expression of the genes. 13 gene expression datasets were used to test GNet-CAC and to compare with other tools that have similar merit of ranking or grouping, such as CogNet, MATE, and SVM-RCE. As illustrated in Figure 2, GNet-CAC performs similar with MATE, CogNet, and SVM-RCE for different datasets. As illustrated in Figure 3, it uses fewer genes than SVM-RCE. Consequently, a biologist may want to discover significant pathways and microRNA targets of these genes together in the same data. In this respect, GNet-CAC, CogNet, MATE, and SVM-RCE integrated biological information into the machine learning algorithm and help wet lab scientists to understand disease mechanisms at the molecular level and to generate hypotheses. Thank you for listening.
Okay, is there any question? So if we don't have from our audience, Fatma, I can ask you that it's not so easy to read the poster, unfortunately, but as I read, at least, you say that 13 human gene expression data sets were downloaded. So how did you decide which 13 that you're going to use? What was the criteria to collect these 13 data sets for your algorithm, for your scoring and grouping, let's say?
Actually, we don't have an criteria, but we looked previously studies.
Sorry?
We looked previously studies.
Mm-hmm. Okay, thank you. So is there any control group or something that you compare with the other scoring? Because there are also similar gene expression based grouping and scoring methods. Did you compare?
Yes, we compare them and
what was the difference, the main difference?
Main difference is they use a number of genes. Our methods use less than number of genes from the other tools.
So you think that is an advantage?
Yes, because
okay, then. You can complete because
no, thank you.
Okay, then. Thank you for your presentation.
Besti, could I comment? Just a quick comment.
Yeah, look, please.
Sure. So Fatma, yeah, for your presentation would have been good if you could have zoomed into the figures, because it's really difficult for us to make sense of what's being shown. So you could have zoomed in, moved around, but it was really static and it was really difficult. So I hope in the future, you could do that when you record the presentation. Thank you.
Yes, thank you. I am sorry, I didn't realize it.
Okay, then we can continue with our next presentation.
Okay, the next one is prediction of antihepatitis C peptides with machine learning algorithms.
Respected scientists, faculties, and my dear colleagues, I, Akash Saraswat, PhD scholar from BML Munjal University, India, would like to present my poster titled Prediction of Antihepatitis C Peptides with Machine Learning Algorithms. This work has been done under the guidance of Dr. Bipin Singh and Dr. Arijit Mehta. Bioactivity prediction from sequence information of peptides is a relatively new field which has gained momentum in the last decade. Various machine learning models have been developed to predict the bioactivity of different classes of peptides. However, there is a limited understanding about sequence and structural features' importance for bioactivity in different peptide classes. In this research work, we have made an attempt to dive deep into the understanding of correlation between sequence-based descriptors and bioactivity of antihepatitis C viral peptides. For the following analysis, we took 420 sequence of well-documented antihepatitis C viral peptides as a positive class, and special considerations were given while preparing the negative data set, which comprises of another 420 sequence of peptides from antiparasitic, toxic, antifungal, drug delivery, and antihypertensive class. Based on the literature review done for this work, decision tree have been described as a significantly improved method of analysis. We therefore used random forest and J48 decision tree for our analysis. Descriptors have been calculated keeping into consideration the maximum coverage of information. For composition-related info, we used amino acid composition and enhanced amino acid composition. The amino acid composition talks about frequency of each amino acid in the sequence, whereas enhanced amino acid composition calculates features based on a sequence window of fixed length which slides. For position-related information, we used composition of K-spaced amino acid pairs, which is a feature encoding that calculates the frequency of amino acid pairs separated by any K residues. Calculation of physicochemical properties is another rich source of information. Here, we have used composition, transition, and distribution features which represent the 13 different types of structural or physicochemical properties computed using the sequence information of peptides. All of these descriptor classes have been well established in literature to predict peptide-based bioactivity information. The metrics commonly used to assess prediction performance of such models include sensitivity, specificity, accuracy, receiver operating characteristics, ROC curve, and area under the ROC curve, and many more. Evaluation of these parameters with 4 different sets of negative data set using random forest algorithm yielded an average of 93.07 as specificity value, 90.96 as prediction value, 0.92 as AUROC value, and 0.92 as AUPRC value within the different descriptor classes. We also prepared an ensemble model of these descriptor classes, which performed slightly better than the individual descriptors as expected. As we have got some encouraging results, the J48 decision tree has been developed to find specific descriptor classes feature of importance for our further analysis. As part of future work, we will be exploring these decision trees to establish cluster based on descriptor class. That is all for my presentation. Thank you everyone for your time.
Thank you for this great presentation. I think you described everything very well, but maybe only the missing part is that how we can use these predicted antihepatitis C peptides. So what is the biological importance of that?
As this is a very limited study, am I audible? Am I audible?
Yeah, we can hear you.
Yeah.
No.
As this is a limited study which we have just started, the idea was we want to explore it further into the mechanism involved in the work. So this would just be another model that is already being well established, and but then we wanted to make it further more integrated platforms with more mechanistic information. So considering that factor, we did not develop a web server as such, but in due course of time, as one of the research that we have planned, we will be further investigating detailed into the molecule, trying to correlate this with the molecular data as well.
Okay, thank you. Thank you for your answer. Probably you have more answer, but do we need to continue, Nihan? You are also checking the time.
I have a quick question. That's
yeah. Then we can.
Okay, thank you. Thank you, Akash. So yeah, good overall overview there. Did you, I may have missed it, did you compare with other existing predictors? How did it compare?
No, sir, we have not made any particular comparison. We have just made our results in reference to the AURC and the other different parameters that I have stated. The idea was that 0.95, usually all the algorithms that are coming up with the results, they are having a very good sensitivity or accuracy scores. So the comparison would have been on an equal level. So we have not done the comparison as of now. And plus, the second part to that question would be, we have not published this as part of a server, web server, because there is another, this is just a small fraction that we have done so far.
Okay, thank you.
Thank you, sir. Thank you, sir, for asking the question, and thank you.
So there is one more question on chat, but maybe Akash, you can type on chat because of the time limitation. We can continue.
Thank you, Nehan. Yeah, definitely, I'll do that.
Okay, then. So the next presentation is IPNMAP and RedMax hash as bioinformatic tools for better interference of RNA secondary structure using experimental probing data.
About these two tools in bioinformatics that allow to infer the structure of RNA using experimental data. So why are we interested in studying RNA? RNA is a small molecule that could be synthesized biologically, could be used in nanotechnology. It could be used in the new generation of vaccines, such as the mRNA. This is what we're having right now for the COVID could have be also used as a therapeutic RNA. It has several functions as a messenger, translation, and most importantly, it has an important role in regulation. So the ribosome activity depends heavily on the RNA structure, the folding of this RNA. And for some retroviruses, the genetic code is encoded into RNA, not in DNA. The key point here is that to understand the function, we can use the structure. Okay, so far we have very sophisticated combinatorial algorithms that allows efficient in silico prediction for the structure. So when we talk about the structure, we can define it in three levels. We have the primary structure, that is the sequence. We have the secondary structure, that is a planar representation of the tertiary. And the tertiary, this is how the RNA fold in the nature. So most of the time when we're talking about prediction, the focus goes to the secondary for two main reasons. The first is that it's very complicated to use dynamic programming for tertiary, while dynamic programming is the basic for all the computational methods. And the second reason is that we can contend with the secondary to understand the function of certain RNA. The other key point in the title was probing experiment. So the probing could be defined as the process of using experimental protocols with a set of RNA replicates to generate a certain structural pattern. It starts with a modification. In this scheme, we have a chemical modification. It consists into incubated probing regions with several replicates of RNA. And this probing region will selectively interact with some specific nucleotides. In this case, they are accessible and they are unpaired. After this, we have a step of reverse transcription where a specific reverse transcriptase is used, and it will translate this interaction region with nucleotide either into a stop, which is the case here, or into a mutation. After this, we count the number of mutations and the number of stops, and we do sequencing, after doing sequencing, of course, and then we got our reactivity profile. And here you can see that we have some peaks corresponding to nucleotides that are likely to be accessible. I want to mention that at this step, we don't have any a prior knowledge of the structure. So we just plot the structures to give you an idea where the interaction could happen. So once we get a reactivity profile, that is an assessment of the accessibility at the nucleotide level, we use it to bias the Boltzmann ensemble, and then to infer the structure that better presents this reactivity. What we can see here is that for some regions we don't have data. And this is one of the main issues of reactivity profiles, is that because of the complexity of the structure, some nucleotides are not accessible, so we don't have data for them. So far, we have a bunch of experimental probing methods. They could be categorized into enzymatic-based method, small molecule modification method, and cross-linking proximity ligation method. So the most known are the small molecule modification method, shape map, DMS-seq, and shape map, and the DMS map, sorry. In 2020, we had this new generation of probing experiments, the lead-seq, the kit-seq, rig-seq. So if you are interested into learning more about computational methods that use experimental data to predict either the secondary and tertiary structure, I highly recommend to take a look at this review. Okay. So I said that one of the problem is that with the reactivity is that we have this partial knowledge. So one of the direction would be to use multiple probing experiments, and it has been first introduced with Cordero and all, where they aligned different reactivities. Then they got the reactivity average, and then they infer the structure for it. We have also this differential shape introduced by Weeks group. And we have also this mutate and map approach that is based on mutating a sequence, then getting the reactivity, then building this matrix of reactivities. So far, we had this. There was a need to find for probing data integrative method to assess the whole folding landscape in order to find structures that are at the intersection of different sources of experimental data, assuming either agreement or complementarity. So I'm going to introduce APBioNet, that is our first approach here. So takes as input a sequence and a set of reactivity profiles, and for each reactivity we build a pseudo-Boltzmann ensemble that contains structure compatible with this specific reactivity. Okay, so in this case we have 3 reactivity profiles, and we were able to generate 3 pseudo-Boltzmann ensembles. Then we put all these ensembles together. The next step would be to cluster these ensembles in a way to find the cluster that contains structures that best represent all the experiments. So we hope that it will represent all the experiments, and also that contains structures that are thermodynamically stable. So one of the applications of APBioNet was published in NAR, and it concerns this RNA, the labeled capillary ribozyme from the genome. And for this RNA, we had 16 reactivity profiles from different experiments. We have the map, we have the capillary ectophoresis with the shape, with shape and with other DMS, etc. And here you can see that when we look at the ensemble distance, this is PCA for the ensemble Boltzmann pseudo-ensemble distances, we see that we have a big cluster that is forming in here. We have another cluster with the CE experiments, and also we have these 2 singletons. So should we consider this singleton or not? It's a big question. So when we do the clustering for the different experiments, we realize that we have these groups from A to H, and the next step was to find the best combination that allows to have a good model. Okay, a good model means a model with high prediction accuracy. So what we did is that we choose experiments across the different clusters, then we build combinations. And the one that gives us a prediction accuracy of 0.85 was this combination. So APBioNet is robust in our knowledge the first integrative method to find structure at the intersection of different pseudo-Boltzmann ensemble. And one of the improvement direction would be towards the validation on multi-stable RNA, such as riboswitches that alternate between structures. So next, I'm going to present the other approach, RedMax, that is specific to DMS map data. DMS actually is supposed to interact with adenine and cytosine, but we have found that paired nucleotides, ECGU, are likely to be modified. And the intuition is the following. So when a DMS molecule hits a nucleotide that is involved in the pairing, the other nucleotide became accessible. Okay, so other DMS molecule came and interact with this molecule, but not only this one, also the one that is in the neighborhood, for which we had developed the more neighborhood for a base pair, and we have also proposed the more neighborhood for a helix. So RedMax takes as input a rare mutation data per nucleotide pair and an RNA sequence, for which it does generate maximal helices, and then we compute the more neighborhood, and then we compute relative differences, and it ends up with sorting helices based on their relative difference mutations. Okay. So this is the output that we have for the 5S ribosomal RNA. So this is the native structure, and it seems that when we do clustering, the first cluster does contain already 2 long helices. So we also tested it for a longer RNA, RMRP in cell, and we were able to get the pseudonode. So RedMax is a data-driven method, a free energy helix-based model to infer RNA structure information. It makes use of a new correlation metric and assumes a strong correlation between interaction bases. Highly DMS-supported helices are likely to be in the native structure. So this work couldn't be done without the involvement of many people, whom I would like to thank, and thank you so much.
Well, thank you for this great presentation. Actually, I really enjoyed. And is there any question from audience? So probably not. So thanks again for your presentation.
Basti, I just want to echo your sentiments. I think this was a great presentation, very clear, and thank you for the wonderful work.
Thank you so much. Thank you.
Okay, so we can continue with our next presentation.
Okay, so the next one, differential expression, alternative splicing and interacting networks of Kcnm A1 in Bruneian breast cancer patients.
Assalamu alaikum and hi, I am Salma Tariq, a PhD candidate from University of Brunei, Dar es Salaam. I'm here to share some of the preliminary finding of a research project that is designed to investigate the differential expression, alternative splicing, and interactive partner genes of Kcnm1 in breast cancer tissues, specifically looking across different histogrades and molecular subtypes of breast cancer in Bruneian patients. Statistically, breast cancer is one of the leading causes of cancer death globally, with high mortality rate due to lack of suitable biomarkers. Kcnm1, a gene that codes for alpha subunit of BK channel, is considered as potential biomarker due to its highly alternative spliced nature that results in diversity of its function. The upregulation of BK channel has been linked to various cancers, including breast cancer, who postulated the idea that alternatively spliced Kcnm1 variants play a role in cancer progression. Therefore, these transcripts can be used as biomarkers to monitor breast cancer progression as well as therapeutic targets. Figure 1 shows a few of Kcnm1 transcripts. We started our research by retrieving the breast cancer tissue samples from Repas Hospital in Brunei and ran RNA sequencing by following the Mana-Trusik RNA exome protocol. After getting the FASTQ files, we performed some quality control and aligned the read to reference genome using splice-aware aligner named as HiSet2. Transcripts are assembled and merged using StringTie, a highly efficient assembler. A statistical package in R called Balgown and isoform switch analyzer were both used for differential expression analysis and visualization of transcript structure. After getting the list of differentially expressed genes, string was used to find the interactions among them. And then cytoscape was used to find the interaction networks of Kcnm1. Initially, we have analyzed 12 breast cancer tissue samples. These samples are divided into different groups, which are control, grade 2, 2+, and grade 3 based on different histological grades. Our preliminary analysis has identified 1,172 DEGs in grade 2 sample, 1029 in grade 2+, and 1048 in grade 3. In figure 3, we have heat map graphs that summarize the gene expression of top 2,000 genes across different histogrades. Genes with expression value below mean are presented in blue color, whereas genes with expression value above mean are shown in orange color. From there, we have found 27 DEGs that are common among all histogrades as shown in figure 4. Other than that, we have found several Kcnm1 transcript interacting genes to be significantly expressed. 27 DEGs in grade 2 samples, 14 in grade 2+, and 15 DEGs in grade 3 samples. On top of that, the DEGs found in tissues of different histogrades were also different. This suggests the potential involvement of different genes in the invasiveness of breast cancer. Top 10 DEGs interacting with Kcnm1 can be seen in table 1. Many of the identified DEGs such as COM3, DPP6, Kcnb3, Kcnb2, and Kcnh5 are involved in the centrosome cycle and regulation of the ion channels. Our individual analysis of sample for Kcnm1 transcript expression shows different transcripts being expressed in different breast cancer samples. So one of the sample S3 was shown to express 7 different isoforms of Kcnm1 variant, as shown in figure 5, with a few of the isoforms being more differentially expressed than the other. 2 Kcnm1 transcripts, Kcnm1, 274, and 242, are identified to be significantly expressed in grade 2 and 2+ samples, as shown in figure 6. The same transcript, in addition to the 2 microprotein variants, are identified as differentially expressed when analyzed with isoform switch analyzer, as shown in figure 6. These microproteins may be involved in the breast cancer progression and development. From this point further, comparative studies on these DEGs involving high number of normal and breast cancer tissues will be needed to provide evidence of Kcnm1 involvement in carcinogenesis and its true potential as a breast cancer biomarker. That's from my side. Thank you for listening to me. Goodbye.
Okay, thank you for the presentation. So it was a little bit fast to catch some points. And I really didn't realize that you talk about Bruneian breast cancer patients. So when you say Bruneian, is it about a diet that you are consuming or directly for the location?
It's for the location. It's for Bruneian breast cancer, basically from Brunei.
Okay, then it was, so it's not a subtype. So it's, I'm not familiar with the use, like we are not saying Turkish breast cancer patients. I'm not using this like breast cancer patients from Turkey. It sounds, you know, something that maybe I didn't catch. So that's why I'm asking directly. So in this location, you are saying that Kcnm1 is most common alternative stabilizing. So is it the only one? Or did you just collect this one?
We are just focusing on this one.
But there are many.
Yeah.
Am I right?
Yeah, we are basically focusing on BK channel. So this is one protein that is affecting the alpha subunit of the BK channel. That's why we are just focusing on this gene in this project.
Okay, then. Thank you. Thank you so much.
So the next poster presentation is effects of protein-protein interactions network statistic in the scoring the pathogenity of single nucleotide variants.
Hi, I am Kcnm. I'm working as R&D engineer at IDEA Technology Solutions. With this work, we aim to measure the effect of the protein interaction for predicting the pathogenicity of the variants. Non-synonymous single nucleotide variants occurring in the coding region can lead to the amino acid change that plays a huge role in various diseases. The general approach is to make a prediction based on the features associated with the variant itself. However, the inclusion of biological processes can be effective for the pathogenicity prediction of variants in the coding region. For that purpose, we extracted the features related to the biological effects using protein-protein interaction networks. We collected the raw dataset from HumSavar and ClinVar databases. We selected only high-confidence curations, for example, manual curation in HumSavar or the variants that have more than 3 stars in the ClinVar. We combined the features from 3 different resources. Firstly, we collected sequential features such as conservation scores, allele frequencies, GC content, amino acid transition probabilities, etc. We also believe that the characteristics of the gene region in which the variants are located can be crucial for pathogenicity. For this reason, we collected the features related to specific gene regions from NOMAD constraints as our second feature resource. The tolerance of intolerance level of a gene for given recessive mutations, the frequency of occurrence of different types of mutations in a gene can be an example of the features we can obtain from this dataset. As we mentioned earlier, non-synonymous variants are the variants in the coding region of the genes and are responsible for protein coding. We can extract useful information from protein interaction networks to find how important the protein is for some biological events. In a protein interaction network, each node represents a protein. Ages are some specific relations between the proteins. The features related to protein can be extracted using the protein interaction networks. The protein-protein interaction network was downloaded from HumanNet version 2, visualized via Cytoscape and analyzed Python, CDLib, or iGraph libraries. To evaluate the impact of the PPIM for prediction, we used the network statistics such as degree, closeness, betweenness, radiality, so on and so forth. We extracted these features in 2 different ways. First, we calculated those features globally. We looked at these statistics for the whole network. Also, to evaluate the effect of clustering and network statistics in local communities, we evaluated clustering methods in the Python CDLib library. Our aim in clustering is to find close relations between the proteins and extract features from these local relations. The extracted features from here merged with the sequential and gene-based features. In the training phase, we used 63,500 117 variants with 140 features. Our training set has 31,285 variants. We dropped constant, quasi-constant, and non-informative features based on different feature selection techniques. The final model was trained with 94 features using 63,517 variants. We achieved 93.76 AUC ROC value and 70.68% MCC for the independent test set. We evaluated our features with the permutation importance technique. When we look at the feature importance graph, all PPIM features have higher importance than the median importance value. The horizontal line shows the median value in the figure. The degree is the most important PPIM feature, and three PPIM features, degree, stress, and between centrality, are among the top 10 features. In addition, many features from nomad constraints have higher importance than the median values. These results show that PPIM and gene-specific features are useful metrics to predict variant pathogenicity.
Hello, I am Rim Shaib, PhD student.
Hi.
Okay, thank you for the presentation. Is there any question? The poster of this effect of protein-protein interactions network statistics. So it was really hard to follow because your presentation is too small. Maybe we need to zoom in some parts. But I have a question that, so you say that while you are discovering this network or investigating the network, let's say, you say that you use different network topological features, right? So when you check your final results, what were the differences between local and the global metrics when you use? What is the main difference between them, or is there any significant difference that you check?
Hello, I want to answer the questions because our main author here didn't attend. As co-author, I will try to explain everything. Okay, first questions. We tried to mention global and local graph statistics, but we saw that okay, with some measurements, some values may differ, but the correlation in them are really high. So we saw that that's why we didn't add the graph statistics of local or global clustering evaluations to the final network. So that's how the correlation was really high. Can you hear me?
Yeah, but I couldn't. Yeah, I was hearing your voice. But I couldn't clearly hear what you were saying. So maybe you can write down, because there are also more questions than I expect now. Did you use feature selection methods other than permutation importance? And how many features did you see in total? Probably it's coming to you. So you can also answer these questions now.
Actually, at the start, as I said, Kcnm is the main machine learning engineer of this project, like a bioinformatics part of the project that I will try to answer fully. We'll start actually, as I explained, we have high number of features in the beginning, but we used feature selection methods in the beginning. Actually, this model we are still trying to improve in some manner, but at first we used feature selection methods because each variant is in the perspective of annotation. There are many features. Sometimes it's really hard to implement a machine learning model with the high number of features. In our final model, you can see that all features are, you can see in the graph. Actually, this is the final model. At first, we had more than 200 features for each variant. I hope that.
Okay, if there is more question, then audience can write through the chat box as well. So thank you. So we can continue with the next presentation, which is going to be an oral one, right?
Okay, the next one is advanced machine learning techniques for multilabel medical text categorization.
Hello, I am Reem Shaib, PhD student in Applied Computing at Beji Mukhtar Annaba University from Algeria. A great pleasure to be with you in this video presentation. In this presentation, I am going to introduce an advanced machine learning techniques for multilabel medical text categorization. During this presentation, we start with an introduction, then we explain feature extraction and multilabel classification. Then we describe the used methodology. In the experimentation part, we explain the different experience carried out to obtain this result, and we finish by conclusion and perspective. With the rapid growth of the medical text dataset, most of which are unstructured, reading and understanding the information contained in millions of medical documents is a time-consuming process. Unstructured medical documents are complicated and very hard to handle, but they commonly include detailed information on patient which is valuable, which requires finding a way to organize them. Automatic text classification aims to automatically classify text documents into one or more predefined categories. In recent years, multilabel classification has become a trend in various fields, and it has received increasing attention in text data. However, for some application, objects must inherently be described by more than one label. One of the main problems in classifying medical text is how to extract the relevant features to better represent and classify those documents. For determining knowledge from medical text, the classification algorithm requires an appropriate set of features. In addition, extracting informative features greatly increases the performance of the classification model and reduces the computational complexity. In such setting, the correlation between labels contains valuable information that can be used to obtain more accurate classification models. For this reason, numerous studies have been carried out in this field, and many machine learning techniques have been suggested for medical text classification tasks in recent years. The goal behind this work is to provide a learning method primarily to rapid decision-making and improve the performance of multilabel classification of medical text. Analyze and evaluate the impact of using intelligent features in the multilabel document classification scenario, and take into account the relationship between labels and see their influence on system performance. And we also want to study the behavior of long short-term memory classifier on specific natural language processing tasks. Before detailing the proposed methodology, we define some basic concepts. Feature extraction or feature engineering is a machine learning process that involves the exploitation of raw data. It consists of extracting a list of words from text data, then transforming them into a set of features usable by classifier. Feature extraction method can be divided into two categories. Traditional features based on word counting. These approaches depend mainly on prior knowledge, which are back of word TF-ID, and automatic features based on semantics. These approaches take into consideration the contextual document semantics, which are word embedding, word-to-vec, doc-to-vec, clove, and enum. Multilabel classification is a challenging problem in the field of natural language processing. It is a variant of single-label classification, where a set of labels is associated with a single instance. However, there are other classification issues where each instance can be associated with one or more labels. As the example below shows, a document can belong to more than one categories. For example, in the case of multilabel medical data, the medical document describing the state of health of a single patient may contain one or more diseases, where each disease is represented by a class. Multilabel learning approaches can be categorized into three main families: transformation learning, adaptation learning, and ensemble learning. In this step, we explain the different techniques used and we show the architecture system. First of all, we prepare our data. The preprocessing step consists of adapting 5 processes. Next comes the feature generation step that uses the glove model. Then we organize the data into training data and test data. In training phase or classification stage, we use one versus all, classifier chain, and Lstm classifier. In the test stage, we test the test data by 3 models to predict the labels.
Nihan, the presentation just stopped, but it's not completed.
Here an example for each process. Glove model is unsupervised learning techniques for the vector representation of words. This strategy, it consists of counting the frequency of occurrence of a word in a context, the frequency at which words co-appear in a corpus where similar words are grouped together and different words repair each for form an occurrence matrix. Glove model captures both global statistics and local statistics of data in order to obtain the vector of words. The one versus all strategy is used in the classification stage. It is an intuitive approach to solving the multilabel classification problem. It uses a base classifier or kernel classifier as a decision model. This strategy consists to decompose the multi-classification problem into multiple independent binary classification problem. In this method, the labels are mutually exclusive. It does not consider any underlying correlation between classes. Classifier chain transformed to a chain of binary classification problem, where subsequent binary classifier in the chain is built upon the prediction of preceding ones. This chaining method passes label information between classifier, allowing classifier chain to take. Also, the Lstm neural network is adapted to classify the features generated by the glove model. Long short-term memory network is a deep learning model based on recurrent neural network. RNN repeats the module each time the input receives new information. However, Lstm remembers the problem for a longer time and has a chain structure to repeat the module. It helps to avoid the problems associated with long-term dependence. The Lstm model choice is motivated by the success that has been achieved in recent years. It allows to capture very long-term dependencies between words. Lstm models contain computational blocks that control information flow, with input gate, forget gate, cell state, and output gate. The feature extraction in the textual representation is the most important step. In this work, we use glove technique. Here an example of vector of the word oxygen produced by glove model. The multilabel classification is the last step of our system, has the main objective the classification of the feature generated by glove model, using the one versus all strategy that use logistic regression as basic classifier, classifier chain, and Lstm classifier. To validate our system, we use OH-SUM dataset. It consists of medical abstract concerning 23 categories of cardiovascular disease. In the result part, we carried out several empirical tests to improve the classification accuracy of our system. From the result represented in the table below, we can say that Lstm have more precision compared to the other methods used in this study. In this slide, we show the visualization of the evaluation metric during the training process. In conclusion, we can say classification of medical text is a special case of text classification. In this work, we have presented a multi-label text classification approach to predict different types of diseases in a medical documents using the intelligent feature generated by GLOVE model. Also, we use a several classifier one versus all classifier chain LSTM to reinforce the learning of the prediction model. In perspectives, we want to enrich our system by using a meta-heuristic approach to find the optimal features vector for better represent the textual data during the generation of large-size vector by GLOVE, and developing an algorithm or technique that can handle multi-label and balanced data. Thank you for your attention.
Okay, thank you for the presentation, Rimchai. So there is one question there in the chat box. How to explain that LSTM gave more performance compared to other methods that you use?
Hello. Thank you for
Hello.
question. LSTM gives more precision because this is a deep learning model which is structured in block sequence. This structure allows it to remember the data passed beforehand and to keep the dependence between the words.
So I have one question that I really would like to know. Maybe it's a little bit silly one, but when you are creating the vector, you give an example like an oxygen, right?
Yes.
GLOVE will create a vector to define this word. So what do you use, in which language that you train your model? In English? So your medical text was based on an English words, right?
Yes, in English.
And so what happens if you use another one then? So your results,
the GLOVE model adapt in any language.
Same or not? What do you think? It's the criteria that you change your result.
Yes, we can use the data in any language.
Yeah, and then does it affect your result or your accuracy, your model? What do you think? How are you going to discuss this?
I don't train in other language, just in data, medical data.
Yeah, but in any medical data could be in any different language. So this typing can work. That's why I wonder that, because some alphabets, some words are also different. So I just wonder that, have you ever wondered or tried to understand how the machine learning techniques will answer? I mean, how they respond to this difference?
Maybe in future I test with another language.
Yeah, because the impact of this work is global. So it's not limited to the English. That's why I don't know. Okay, thank you. Thank you so much.
Thank you for you.
Beste, I have a question.
Then we can keep your question, Asif, please.
Okay, very quickly, Rim, what do you think if you were to merge all the three, classifiers into one? Do you think that's possible? And do you think you'll get a better result compared to LSTM?
I use three classifier to analyze the impact of correlation between labels and between feature. For example, in one versus all strategy, we
do you think it's possible to integrate them? Is it possible to integrate them?
Integrate LSTM in classifier chain, for example?
Yeah, all the three, somehow integrate and see if there's any benefit.
Yes.
Okay. All right. Thank you.
Thank you for you.
Okay, then we can continue, but probably you have one other question in the chat box stream.
Yeah. So, okay. So you can type on chat, Rim. Okay. So the next one is co-expression network analysis reveals novel prognostic and diagnostic network biomarkers in cervical cancer.
Hello everyone, my name is Mehdi Kori. I'm PhD student at Marmara University at Bioengineering Department. Today I would like to inform you about our study entitled as Co-Expression Network Analysis Reveals Novel Prognostic and Diagnostic Network Biomarkers in Cervical Cancer, which was conducted by Esra Gök, Kazım Yalçınerga, and me. Cervical cancer is the most common encountered cancer among women after breast cancer. Also, the death caused by cervical cancer is high. So, according to this data, it is clear that the prevention or treatment strategies are needed. In this study, we performed co-expression network analysis. We believe that the gene expression studies should be complemented by differential co-expression studies to map the correlations among the genes, because we know that the genes never function alone and they are presented as connected networks. In this study, we aim to perform the meta-analysis of gene expression datasets and revealed common differential co-expressed genes. Then we performed differential co-expression network analysis using common DEX to find altered co-expression patterns of genes between disease and control phenotypes. Then we performed prognostic and diagnostic performance analysis to co-express gene modules. And our final aim is to discover prognostic and diagnostic modules, which we can say systems biomarkers for cervical cancer. When we look at the methodology that we applied, we first screened the gene expression datasets of cervical cancer from GeoOmnibus. Then we identification of differentially expressed genes of transcriptome dataset individually. Then we identified the mutual differential expressed genes among all datasets individually, and we call them core down and core up regulated genes of cervical cancer. The correlation pattern of each core gene pair in each state were calculated by using Pearson correlation coefficients. Then we identification of positively co-expressed and negatively co-expressed gene pairs. We differential gene co-expression networks were constructed around differentially co-expressed gene pairs. Then by using MCOD plugin, we identified the modules within the constructed networks. Then we identified the significant modules via module size, clustering coefficients, and network density metrics. Then we last preclinical validation of prognostic performance of the modules via Kaplan-Meier plots, and we validate the diagnostic performance of the modules via principal component analysis. When we look at our results, in figure 1, we see a consequence of meta-analysis of differential gene expression analysis. We found 130 down-regulated core DEX and 199 upregulated core DEX. By using these DEX, we find differentially co-expressed gene pairs, and according to Pearson correlation coefficient results, we constructed 4 different networks. And by using MCOD plugin, we found modules from our 4 different networks, and as a result, we find 7 modules. The gene pairs of modules 1 to 6 shows positive correlation in healthy state and no co-expression in disease state, whereas module 7 shows positive correlation in disease state and no correlation in healthy state, as we can see in the figure 3. In figure 4, we saw prognostic and diagnostic performances of the modules, and the module 1, 2, 4, 5, and 7 have significant results. And we can say that they can be used as prognostic and diagnostic systems biomarkers for cervical cancer. In conclusion, with this study, we offered 5 systems biomarkers which show prognostic and diagnostic capabilities for cervical cancer. Thank you.
Thank you for the presentation. So what can we do after that? after you define these modules, Mehdi? What is going to be the next step?
We can try these modules by experimental studies if they really show diagnostic and prognostic capabilities in the cervical cancer.
So do you offer to use a method like a qPCR?
Yes, maybe.
To understand the difference as a model.
Yes.
Is there any question?
Yeah, I have a question.
Please.
Yeah, as she said, he has used meta-analysis. So I have a question that how did he normalize data, and how he removes batch effect and other incorrect data from which take data in her study?
For the differential expression analysis, we use R platform, and the R platform we use EPI package, and we use RMA normalization method actually.
Okay, thank you.
Thank you.
Now we can continue with the next presentation.
Okay, then. Prediction of linear cationic antimicrobial peptides active against gram-negative bacteria based on machine learning models.
Hi everyone, my name is Ümmü Gülsüm. Today I will explain our project. Our project title is Prediction of Linear Cationic Antimicrobial Peptides Active Against Gram-Negative Bacteria Based on Machine Learning Models. First of all, what is antimicrobial peptides? Antimicrobial peptides are considered as alternatives to the conventional antibiotics. So prediction of antimicrobial peptides are important because it is an efficient alternative to experimental methods, and thanks to prediction of antimicrobial peptides, we can create novel drugs. So it is important actually. In our project, first of all, we create a data set, and this data set we create according to some rules. First of all, from the BASP web server, we select peptides that active against gram-negative bacteria, and then we select linear cationic peptides between 20 and 50 amino acid in length, and then we select non-hemolytic peptides, and after we download these peptides, we filter these peptides according to similarity. For this purpose, we use CD-HIT program, and we select peptides that is similarity range is 8%. And then we assign our class labels according to minimum inhibition concentration values. If our peptides having some MIC values smaller than 25, we assign this as positive, and if it is bigger than 100, we assign it as negative. After this, we create features, we export features, and our features are these 10 features: sequence length, normalized hydrophobic moment, normalized hydrophobicity, net charge, isoelectric point, penetration depth, tight angle, propensity to disordering, linear moment, and propensity in vitro aggregation. And is this table an example of our AMP and antimicrobial peptide and non-antimicrobial peptides included in our gram-negative data set. As you see, the first column is sequences, and the other 10 columns is our features. The other is our minimum inhibition concentration values, and according to these values, we create our class labels. Then we construct our model. We separate our data set as training set and test set. 90% of our data set as refer as training set, and 10% as refers as test set. Then we applied classification models, machine learning models on training set. These models are AdaBoost, LogitBoost, decision tree, random forest, support vector machine, and these two stack learners. Then after we trained our model, we applied our test set on this trained model. and we evaluate our performance metrics. And these are our performance metrics that we used in our project. After we applied these models on our data, we take our results. When we look at our results, we can see that random forest gives best results when we compare to other models. And also not only for accuracy, also for other metrics, it gives the best results. And as a conclusion, in this study, we attempt to develop a robust classification model for antimicrobial peptide prediction. And we have compiled our dataset from the DBASP web server. And at the end, we can say that the random forest algorithm achieved better results when we compare to other algorithms. And this prediction approach might be useful to evaluate the antimicrobial potential of a candidate peptide sequence before experimental studies. Thank you for listening. If you have any question, I will be glad to answer.
Thank you for the presentation. Is there any question? So probably
Leslie, I have a question.
Yeah, please.
Okay, thank you, Umi. Sorry, can't read your name. Anyway, thank you for the presentation. And I have a question. It seems like a lot of studies are observing random forest to be the best performer. And for example, I'm not sure if you used SVM, but I wonder whether it's really because by default random forest is best performer. But if you were to optimize the parameters of all the others, you might actually get something comparable. There's a lot in the literature about SVM being very good, but you don't really see that unless you optimize the parameters. So I was wondering, did you use SVM? And if you didn't, for the other methods that you use, did you actually try optimizing the parameters, or you just use default parameters?
Thank you for question. We use separate vector machine, of course, and before applying this, we also optimize our parameters. These are also parameter optimization results.
Okay, so this is so you're saying despite optimization, you still got random forest to perform better.
Yes.
Okay,
yes.
That's interesting. Thank you.
Thank you.
And there is another question. When you eliminate sequences with a similarity cutoff 80%, is not there a possibility that you lose a probable cation peptide for antimicrobial activity?
What is not the possibility you lose a probable cation? I think no, because also after this work as a future work, we want to find a motive. And so we try to eliminate similar peptides for this purpose. We don't want to put similar peptides format findings.
Is that an answer to your question, Deepika? So probably we're going to be discussing this through the chat box. Okay, so let's continue with the next presentation, which is going to be an oral one.
Okay. Decoding molecular signatures of pituitary neuroendocrine tumor subtypes through systems biomedicine perspective.
Hello everyone, I'm Büşra Aydın from Konya Food and Agriculture University, and in this talk I will present the results of our latest study, which is decoding molecular signatures of pituitary neuroendocrine tumor subtypes through systems biomedicine perspective. First, I will briefly mention about the systems biomedicine. It is a sort of application of systems biology approaches for the benefit of medicine. And it is an emerging discipline now, and it integrates many areas such as medicine, systems biology, molecular biology, bioinformatics, etc. And in the era of high throughput technologies, by the investigation of large-scale screenings and clinical data, specific biomarkers, novel therapies, prognostic assessments, and uncovering of molecular mechanisms can be possible. Let's talk a little bit about the disease we are searching on, which is pituitary neuroendocrine tumors. I call them PITNET hereupon. And pituitary is bean-shaped tiny structure that's situated at the base of our brains, and it has two parts as anterior and posterior. These parts secrete different hormones such as growth hormone, prolactin hormone, adrenocorticotropic hormone, follicle-stimulating hormone, luteinizing hormone, and thyrotropic stimulating hormone. And sometimes these hormones can be overproduced and can cause PITNETs. These tumors are non-metastatic neoplasms of the pituitary and considered as a rare disease. But the number of patients diagnosed is increasing day by day, and there is a tremendous unmet need and opportunity to discover new therapeutics for PITNETs. Although there are many drugs that have been used for the treatment of PITNETs in the clinic, many of these drugs have side effects, as all you know, chemotherapeutics, and show different outcomes in context of the total recovery, considering tumor recurrence and relapse. At this point, systems biology approaches enable the integration of multi-omics data and the discovery of novel therapeutics and treatment strategies through techniques such as drug repositioning. While mentioning on drug repositioning, I want to say a few words about DR. Drug repositioning, also called drug repurposing, reprofiling, or retasking. And this strategy is known as all drugs for new uses, and it's an effective strategy to find new indications for existing drugs. Drug repositioning has become a popular strategy in recent years, since they all are approved drugs. They are accepted as highly efficient, low cost, and riskless. DR can be carried out in three ways: activity-based, knowledge-based, and in silico-based. You can see the drugs that were repositioned out of their indications recently. For example, minoxidil is an hypertension agent, but lately used for hair loss. Back to our study. To search the PITNETs at transcriptome level, we collected 9 PITNET tissues, including subtypes of non-functioning and functioning PITNETs from Marmara University Faculty of Medicine, Department of Neurosurgery. And then we isolated the RNAs of these tissues with a nucleosome RNA kit isolation kit. Then library constructions were carried out by AmpliSec kits purchased from Illumina. Then samples were sequenced by Illumina Novasec 6000 device with reading length of 1000 base pair paired ends and reading depth of 20 million. In single, after that, we quantified abundances of transcripts from RNA-seq data with using Callisto program. After elucidation of TPM counts, differential expressed genes were determined using DSEC2 package and using our program. Enrichment analysis of the DEGs were performed using Metascape and Consensus PathDB online tools. Then we elucidate reporter molecules such as microRNAs and NTFs using a reporter features algorithm. Then we constructed 3-level biological networks, including transcription factor target DEG networks and microRNA target DEG networks and PPI networks. Interaction data were retrieved from BioGrid, Multilabel, and HDRI databases. Then we performed drug repurposing analyses using hubs of these 3 networks. When we look at the results, heat map showed top genes sorted by their adjusted p-values and log FC values. It can be seen that these genes can divide samples into two groups as functioning and non-functioning samples. Bubble plot at right side represents the 6 genes with the lowest p-values, and each of them can cluster functioning and non-functioning tumor samples also. The volcano plot at the left side represents the differential expression of the genes. At total, we have 941 genes. We performed functional enrichments of both up and down regulated genes. For upregulated tags, biological processes such as synapse organization, synaptic signaling, neuron migration, and regulation of neuron that were enriched. For downregulated tags, corticotropin-releasing hormone signaling, EGFR signaling, MAP kinase signaling, proteasome degradation were the biological processes that were enriched. We also performed PCA analysis with using these DAGs and found that DAGs have potential to cluster major types of pitnets as functioning and non-functioning samples, and can cluster also functioning types of pitnets such as growth hormone secreting, adrenergic-corticotropic hormone secreting, and prolactin secreting samples. Then we constructed PPI networks around our DAGs with using BioGrid's latest release and visualize these interactions via cytoscape. The determination of hub proteins was carried out via CytoHub plugin. Hubs were elucidated using degree and between the centrality metrics. You can see at the right side the hubs of this PPI network. Same procedure were followed for microRNAs and microRNAs that were collected from Mirtar-based database. And microRNA DAG targets interactions can be seen in the left side. At the right side, hubs of microRNA DAG target network are presented. Also, experimentally validated transcription factors and their DAG target interaction network was constructed. The information retrieved from HTRIDB database. Hubs were also elucidated for this network. After construction of biological networks, we use these hubs elucidated from 3 biological networks as query for drug repositioning, employing links data by using L1 CDS square search engine. Then we got these drugs. Some of them has a role as antineoplastic agents such as galvanomycin and salarmide, or antifungal agents such as ripostatin A. And we have 2 histone deacetylase inhibitors, Verinostat and Categorization A. The approval status of the drugs can be seen in the table. And except galvanomycin and Verinostat, all of these drugs were not reported before for treatment of pitness. Galvanomycin and Verinostat were reported also by our research group. As a conclusion of this study, I can say that RNA-seq analysis of pitnet samples, including different subtypes, was carried out to elucidate pitnet-associated genes and their biological networks, including proteins, transcription factors, and microRNAs. There's a potential to cluster functioning and non-functional pitnet types, and might lead to design effective therapeutic strategies in disease prognosis. Our findings are based on in silico analysis, so a critical extension of this work would be to learn whether these patterns can be recapitulated by in vivo or in vitro experiments. All in all, future efforts should be carried out to incorporate these systems-level understanding of proposed genes and potential therapeutics into clinical practice. I will finish my presentation with this slide, and thank you for listening. If you want to contact me about this study or anything, this is my email address. You can reach me about this way, and thank you.
Thank you so much for this nice presentation, Bushra. Is there any question? Okay, if we don't have, we can continue. Asif,
I have.
probably you have.
Thank you, Vesti. So, Bushra, wait. So, okay, Bushra. So thank you for the presentation. Very nice, very comprehensive. So I really saw that you went out and you did so many different things. In terms of the validation, where are you going to be actually carrying out this, or is it just going to be a stop here and you're hoping somebody else would pick it up and do the validation?
Okay, thank you, Asif, for your interesting question. This is the first part of our study, and we will continue with the experiments with using cell lines first. Then, if we have promising results, we will continue with the in vivo experiments.
Wonderful, thank you. And I'm noticing that a lot of this drug repurposing work, utilizing or maximizing on the networks approach. So there seems to be really something there, and that seems to be the direction people are going. So congratulations, and hopefully you get something promising.
Thank you, thank you.
Bushra, you didn't say, but its projects also supported by TUSEP. So it's very much knowledge.
Yeah, I understand.
I also one of the partner of this project. So that's why I can say that we are going to be going further to analyze what we have found. Okay, Bushra, we have one more question. So how many reads per sample did you get from your sequencing?
As a reading lens, we used a 100 base pair reading lens. Am I understand correctly your question?
Then
we use 20 to 4 million per sample reads. If you okay.
It was in the slide actually.
Thank you.
Okay, perfect then. Probably we are done. Now we can continue with the next presentation. DCNA. You can.
DCNA, a new tool for differential co-expression analysis of gene expression data.
Hello everyone, I am Yasir Demirtaş. Today I will make a poster presentation about DCNA web-based tool. DCNA is a web-based tool that provides a new pipeline to perform differential co-expression analysis and constructs network using a different group of gene expression data with novel methodology in the background. Differential co-expression analysis aims to identify gene pairs that behave distinctly under different conditions by computing gene expression correlations and making statistical analysis of multiple samples. The advantage of the network-based methods is extracting meaningful information from complex data. Also, visualization makes it easier to understand data. With network biology methodologies and concepts, differential co-expression networks have become popular to understand the molecular mechanism behind the disease or specific research subjects. Via topological assessment, offer valuable knowledge regarding biological alterations based on gene expression by comparing two context-specific conditions, such as disease and healthy tissues, or chemical applied and not applied situations. Finding gene correlations in human disease is important for different group studies. Some of these studies, finding diagnostic or prognostic biomarkers, classification of disease and disease stages, or monitoring response to treatment. However, commonly most of the differential co-expression tools are are language-based. For this reason, researchers must be able to use the R programming language to create differential co-expression network. This is the main starting point of this study. We wanted to develop a web-based tool for researchers that can easily analyze their data without dealing with any programming language. I want to talk about tool algorithm. First of all, we have applied z-score normalization to gene expression values. Afterwards, normal distribution was checked in disease and healthy tissues separately. Pearson correlation coefficients and Spearman correlation coefficients were calculated for situations. With these coefficient scores, cutoff was determined to identify statistically significant pairwise correlation of genes. Finally, we filter out significant gene pairs with use these cutoff. In the differential co-expression part, we have two parameters. First parameter, it includes gene pairs which is significant in disease but not significant for healthy situations. Second parameter, it includes gene pairs which is significant for both states, but if their correlation score difference is bigger than 0.5. After talking the algorithm, I would like to talk about the technologies involved in the development of dsNA web tool. For algorithm development, we have chosen Python language. For web framework, we have chosen Django framework. For the database, we have chosen MySQL. In front-end development, we have used HTML, CSS, and JavaScript. Finally, in network construction part, we have used Cytoscape and MCode plugin. Cytoscape is a cross-platform and powerful network creation and visualization software. MCode is a clustering algorithm based on the graph theory developed to find connection between protein-protein interaction sections. In the closing of presentation, I want to give a brief information about usage of tool. Firstly, we need to prepare and upload gene expression data, and we need to specify control sample number from the tool homepage. Then tool separates data to disease and healthy and finds significant genes after normalization, correlation, and statistical analysis. And then in the background, MCode plugin with Cytoscape was used to create a correlation network to find highly correlated gene groups. Finally, you will be able to reach constructed differential co-expression network clusters. You can reach tool at dsna.computationalbiology.org. Thank you.
Thank you for the presentation. Is there any question?
Yeah, I have a question, Beste. All right, I looked at your website. It looks nice, except that for the you only at the moment allow for what I think it was gastric. Is that the only one that's predefined? Are you planning to add some more?
Actually, yes, we are planning to add more data, but these are our data. If you can, you can prepare your own data and you can analyze your own data. The main point is you can use tool with your own data, but for the researchers, we added some example data. You can see, for example, for gastric cancer, you can select gastric cancer dataset and you can see the output.
Right, but and did you benchmark with others, with other methods of differential co-expression?
Actually, I am not, because I used the new methodology. I didn't compare with the other methods yet, but I am planning to compare the other methods.
Yeah, so methodology is normal, but at the end of the day, it's always good to know how much better is this compared to the others, so that the real value comes up.
Yeah, definitely.
Anyway,
Yeah, I agree.
All the best.
I agree. Yes, I agree.
Thank you.
Thank you.
Okay, thank you for the presentation again, Yasir. So we can
Thank
continue
you.
with our next presentation, poster presentation.
Okay, then. Competition analysis of the potential impact of MTC complex SMPs associated with male infertility.
Good morning, everyone, and welcome to my presentation. First of all, I would like to quickly introduce myself. I am Hermep Houda, a PhD student from Human Genetic and Genomic Laboratory at Pasteur Institute of Morocco. And today, I'll be presenting the results of my article about computational analysis of the potential impact of meiotic telomere complex missense SMPs associated with male infertility. During meiotic prophase 1, chromosome undergo rapid movement that is the formation of inter-homologous recombination intermediates, underlying synapses, crossing over and segregation processes, and ensure the pairing and eventually recombination of homologous chromosome. The recently discovered tripartite complex comprising telomere repeats binding pocket formation, protein 1 and 2, type 1 and type 2, and membrane and core junction protein MAGAN has been demonstrated to be involved in these processes by tethering telomere ends to the nuclear envelope, transmitting cytoskeletal forces via the link complex to drive this rapid movement and assembling of meiotic mammalian telomeres on the nuclear envelope during prophase 1. Furthermore, disruption of or mutation of any of the MTC genes causes meiotic arrest with loss of telomere attachment and chromosomal movement with the failure of DNA double strands breaker pair and a disrupted synapses. Accordingly, the disturbance of telomere nuclear envelope connection impairs the homologous searching that cause subsequent meiosis defects, including ablated telomere nuclear envelope attachment, aberrant homologous pairing, and disordered synapses leading to male infertility. This study aims to explore or identify the deleterious SMP in the type 1, type 2, and MAGAN genes by predicting the significant pathogenic impact on the function, stability, and structure of type 1, type 2 complex using different and various bioinfrastructures as a systematic in silico approach, such as variant effect predictor, mutabines, MuSiDeep, MSD calculation, conserve, duet, Hadoop and Protege web servers, and leak for protein-protein interaction. The figure 3 shows the mutation classification and consequence type of type 1, type 2, and MAGAN genes. In this study, only missense SMPs or mutation were included. Figure 4 shows the heat map of global binding energy score of the two complexes. Regions with the light orange shades represent complexes with the higher binding energy and lower binding affinity. Figure 5 shows the superimposed 3D structure of the wild and highly deleterious mutated protein, and figure 6 shows the result of conservation prediction of type 1, type 2, and MAGAN protein using conserve web server. Table A shows the summary of the most deleterious mutation generated by mutabines web server. Table B represents the MSD calculation results for type 1, type 2, and MAGAN protein using Yasara web server. Table C shows the stability prediction of the three protein using Duet web server. And finally, Table D shows the MuSiDeep analysis for post-translational modification sites. So different bioinformatics tools were used in this study, and in total, out of 32 mutations, only 11 SMPs were predicted to be the most damaging by at least 7 software and exert deleterious effects on the type 1, type 2, and MAGAN protein structure and likely function. Additionally, they revealed higher binding energy, less binding affinity, a decreased stability, a high conservation score, higher MSD values, and having a role in post-translational modification. So, in conclusion, 11 mutations were predicted to be high-risk pathogenic SMPs in the current study. Combination of multiple in silico tools provided information required to predict the effects of this mutation on the functional and structural deviation of type 1, type 2 complex, and type 2 MAGAN complex. Hence, our results added to previous knowledge and supported earlier findings that may be helpful for further understanding the role of meiotic telomere complex in male infertility. The results highlighted the need to take into account type of protein-protein interaction when characterizing the variant within the complex. And nevertheless, bioinformatics tools cannot replace conclusive experiments, and their results should be verified by supplementary data. And thank you for your attention.
Nope. Thank you for the presentation, Hoda. So do we have any question?
Just a comment, Hoda, that was very fast.
Yeah, I was also thinking that, because you have at least 2 more minutes, so which makes you it makes you a little bit relaxed probably, but
Yeah,
now
sorry.
it was also really hard for us to follow you sometimes. But thank you for the presentation. So
Thank
we
you.
have
Okay,
our
maybe
last presentation of the day, by the way. Are you gonna have anything to say?
just since we have 2 more minutes, she has 2 minutes. I think we should give her the benefit of that speed.
Yeah, then
Yeah. Okay.
Hoda,
So,
if you want to say anything.
Yeah, I just want to ask a question. I mean, I must say it was really hard for me to follow because it was too fast, but I think I want to try to understand. So when you say computational analysis of the potential impact of MTC, so could you kind of summarize the conclusion of what did you, what did you really find out from this?
Yeah, can you hear me?
Yeah, clear.
Okay, so in this study, out of 32 mutation missense SNPs, we found out that 11 mutation can that 11 mutation can really have impacts and effects on this on the malinfertility. Whether by their stability effect, or we use.
You muted yourself? You got muted. Yeah.
So as I said, we use a lot of bioinformatic tools, so we can be sure that this 11 mutation have really effect on malinfertility. By a degree stability, an effect on the function, and also the interaction of protein as the complex, because this 3 protein, TERB1, TERB2, and MAGEN, has 2 complex, TERB1 and TERB2 complex, and TERB2 and MAGEN complex. So if this protein are affected or impacted by whether stability, the function, the structure also, this can have effect on the malinfertility or cause an arrest in the meiotic meiosis process, an arrest in the meiotic the meiosis defect, anabolized telomere and nuclear envelope attachments, also a disrupted synapses. So all of these processes can lead to malinfertility.
And these were based on your own samples, or this is something from the public database? I mean, these 32 SNPs, and then you narrow down to 11. Where did you get that?
No, there are also other studies, but this is based on my own study.
I see. And how big was the sample?
Yes. This article, we just received the correction of this article. Generally, there are not a lot of articles and can be published in 1 month, I guess. But these results were by various studies were confirmed our results, and this study were published earlier in 2018, 2019.
Okay, thank you so much.
Welcome.
Thank you again, Hoda. So
Welcome.
we came to our last presentation of the day.
Yes. Finding new putative genes having role in excess boron responsive mechanism from probe sets identified as unknown in white cultivars.
Hello everyone. Today I will give a presentation entitled Finding New Putative Genes Having Role in Excess Boron Responsive Mechanism from a probe set identified as unknown in wheat cultivars. Before that, I want to give a very brief information about boron. First of all, boron was discovered 1808 by these three people. It's a member of metalloid family elements from group 3A of periodic table. It has properties between metals and non-metals. It's a relatively rare element in Earth crust, representing only 0.001%. And also Turkey and our country and USA are the world's largest producer of boron. And also our country has also almost 72% of world boron reserve. It does not occur in nature as free element, and there are over 200 naturally occurring boron-containing minerals, and boric acid as the main compound exists at physiological pH. And it's essential micronutrient for vascular plants. The requirements of boron vary tremendously among species and even among genotypes within a species. It's relatively unreactive. However, it can form strong complex with few metabolites that have hydroxyl group in the cis conformations such as ribose, apiose, sorbitol, some phenolics such as, and also amino acids such as serine. So the function of boron actually its only function, well-defined function, is a structural function integrity with cell wall due to cross-linking of apiose residue of two Ramnagel electron complex in cell wall. In nature, although lack of boron results in boron toxic disorder, high soil concentration of boron may also be toxic to plants. Under high rainfall condition, it can be easily leached and this causes deficiency in plants. On the contrary, under low rainfall condition, boron cannot be sufficiently rich and therefore may become toxic to plant growth. There are two anthropogenic reasons for boron toxic accumulation. The first one is over-fertilization with mineral high and B, and also use of irrigation water high and B, leading boron accumulation at toxic level in the soil. So actually, there are three main causes have been proposed, taking into account of our knowledge of boron chemistry. The first one, it alters cell wall structure and also disrupt metabolic function by binding ribose moiety of molecules such as ATP, NAD, and ADPH, and also disrupt of cell division and development. So actually, this is one of article title. Actually, until we published this work, overall patterns of gene expression chains and their physiological and biochemical significance under boron toxic condition in sensitive and tolerant wheat cultivars have not been proposed. So in this article, we use map analysis for microarray experiments. This is come from this program. Shows the differential expressed gene in boron sensitive wheat cultivars ATI under boron toxic condition. And red one is the upregulation, indicate upregulation, upregulated genes. Blue one is the downregulated, as you see. And also, this is also boron wheat cultivars. As you see, there are very limited number of the genes showing this differential expressed and both in sensitive and tolerant wheat cultivars. Actually, we use microarray analysis with small number of these, unfortunately, because a large number of the genes that were responsive to boron toxicity were found as unknown in this wheat cultivars. Actually, this is original result from map analysis. As you see in this program, totally in living organism, there are actually 34 different pathways. The last one, 35, is the unknown. It's not assigned unknown group actually. As you compare this one, the large number of these genes indicate, unfortunately, indicated by this assigned unknown prop sets. So if you talk about this number, 268 and also 249 differential expressed genes were categorized as unknown in leaf tissues, ATI and bolal, respectively. ATI is a sensitive boron cultivars, actually, wheat cultivars. Bolal is the tolerant one. And also 376 and 207 differential expressed genes were categorized as unknown in root tissues of ATI and bolal, respectively. Actually, in this work, our strategy to find putatively by using harvest program and also its hyperlink property to other sequence databases such as NCBI, Harvest Blast, and TAIR to obtain Arabidopsis orthologs. And finally, we use some G group Go programs to obtain ontologic analysis. So our Harvest is actually originated as ESD database viewing software, and we use this program for microarray content design, SMP identification, genotyping platform design, comparative genomics, and coupling physical and genetic map. And also this program is developed by University of California. Yes, this is Harvest with chips. We use this search with chip. We upload this Affymetrix code from with cultivars, and by using Blastics for this ESD similarity, we obtain AGI codes of this Arabidopsis thaliana, and we can use this there. This is score number, this is probability level. So by using this program, actually 95 and 73 differentially expressed genes were putatively annotated in leaf tissues of Atay and Bolar respectively, and also 126 and 39 differentially expressed genes were putatively annotated in root tissues of Atay and Bolar respectively. And then we collect all of this orthologs, Arabidopsis thaliana orthologs. We use AgriGo program. This is gene ontology toolkit for agriculture community, and also we use singular enrichment analysis for this unknown gene group. This is result from AgriGo program. Left one upregulated, differential upregulated genes in tolerant cultivars in bolal leaf, and also we use only molecular function. Only one category group is a catalytic activity is represented for this group. On the contrary, in differential upregulated genes in Atay leaf, there are 3 representative groups as a molecular function. The most actually overrepresented one, structural constitutive of cell wall. So if you look at the downregulated in bolal leaf, only binding category are represented. If you look at the downregulation, as you see, there are many number of those groups categorized in molecular function. If you look at the upregulated bolal loops, there are also only one group is a binding. If you look at Atay group, there are more than one group represented in this cultivar in root tissues. And finally, this is downregulated in bolal roots. Responsive stimulus are only one group represented in bolal cultivar in root tissues, and also there are also more than one. Most of them related to binding are overrepresented in root tissues of Atay. So I don't want all of the tables to actually obtain all of the tables, including prop set from which cultivars. This is unknown group. This is our Arabidopsis thaliana orthologs. This is map function from MapMan. This is function from Harvest, default change, and corrected p-value. This is example of only for upregulated, differential upregulated genes in sensitive cultivars. So first of all, I want to thank my graduate and undergraduate students, and also thanks for listening, all of you.
Thank you so much for the presentation. So is there any question?
Best day, I have a question.
Please.
All right, so thank you. I think Dr. Che-Hyun. Sorry if I didn't get it right. Did you consider doing what do you call this pathway enrichment analysis once you got the differentially expressed genes? Did you? I didn't see any results. Why didn't you proceed to do pathway enrichment and possibly some network analysis, biological network analysis of the DEGs that you got?
Sorry if I understand you. Pathway enrichment analysis, you said.
Yeah, like, I mean, you have the genes, like what kind of pathways do these genes actually perturb or affect?
Yeah, after actually gene ontology analysis, we use MapMan program. It's a specific for pathway analysis. Actually, we upload the MapMan again after go on analysis. But unfortunately, MapMan program gives these similar results because they couldn't find these genes again. For this reason, we had to use BLASTX, and we obtain tables according to harvest programs. Actually, we obtained tables one by one, unfortunately, because we couldn't find any specific pathway analysis for wheat, especially.
Well, you may want to look into David, and of course, the better half of David, or the better one would be Ingenuity IPA, pathway analyzer. I'm not sure if they have anything with regards to plant, but something you may want to explore, David and IPA.
Yes, maybe right, but wheat is a highly problematic one of the plants, unfortunately.
Yeah.
Yes, high complex structures, repetitive sequencing. For this reason, our prop set, unfortunately, is highly complicated.
But you did identify the genes, and the genes, I think, could be further analyzed.
Yes, maybe. I think after taking off this Arabidopsis orthodox, my job is, I think,
it's
better.
Easier.
It's easier.
Exactly.
Yeah. You're right. Yeah.
Okay.
Yes. After that, we
All right,
continued to obtain this pathway.
Sure.
Actually, we tried one for map analysis. I think our graph is better.
All right. All the best.
Yeah.
Thank you.
I have also one question, Dr. Cenk.
Yeah.
So you mentioned that you checked the gene ontology terms, and as I remember, again, can we go back to the slides? Maybe Nihan, you can help us. So it was like there was one gene ontology term that I see, protein binding, and it was while you were checking the downregulated on the leaf.
Yeah,
it was there.
Downregulated, yeah, downregulated, upregulated is to independent examines, I think. If you,
Can we check it again? Actually, my question is about how you can interpret this. So there's one set. So when it's downregulated, your gene set, then it affects the protein binding.
Actually,
what is the biological meaning of that?
Yeah. Actually, we published this data by using small number of the genes. Actually, I show in this presentation. Yeah, this one. And actually, we choose molecular function. I think it's better because biological process, it's how can I that this is not examined in more deeply. For this reason, molecular function is better, I think. Actually, our strategy compared to our previous result, because we publish our data by using known genes in wheat cultivars, but this comes from unknown genes. Actually, we try to compare this gene ontology result with our previous published article. Actually, I checked the results. As far as I remember, all of this Go category are highly similar to our published data. It's interestingly. Actually, because during my preparing my articles, I really wonder this unknown gene prop sets. Actually, after obtaining the results, comparing to our Arabidopsis orthodox, actually we obtain similar results among the unknown groups. Actually, in tolerant one, for example, in left one, mostly we found binding category, and also we know this Excel files and MapMan results. Actually, we know which gene groups are related to binding, and also in the right side, in upregulated and Untitled, actually we found more than one category. Some of them catalytic activity, some of them binding. I think this is highly similar to our previous results.
Okay, then. So maybe it's kind of pattern that you face with your samples. Okay,
yeah, pattern is similar inside of the unknown groups. Yeah.
Okay, then. Thank you for your presentation.
You're welcome. Yeah.
So, Nihan, did we complete all the presentation?
Yes, we can. Okay. So thank you, Dr. Beste, and our honorable judges. Please ensure that you fill up your judges form. I would like to thank all our participants for being here with us. We will now return to the main hall, where the next session will begin at 4:10 p.m. Turkish Standard Time for our last keynote talk by Anton Finstra. Thank you so much.
Thank you all for your presentations and participation.
Thank you, everyone.